Afternoon, ma'am.
Yeah, hi.
Ma'am, this is Aranja. Ma'am, I had a doubt regarding the assignment which was given to us.
Yeah.
Uh, in one of the questions, we had to design a primer for a sequence that is 1000 kb upstream of the coding sequence. Uh,
Yes.
yeah, in that question, the problem was that in my particular gene, there are no refseq genes in the database. So I'm not able to find out that 1000 kb gene sequence, 1000 kb nucleotide sequence. So
There will be an option called RefSeq genome sequence.
exactly, ma'am. I
So,
tried doing everything. It didn't
okay.
work out.
No, you what you have to do, put the name of your gene on NCBI and go down. You'll see a option of genomic sequence. You tap on that, then you will see on the right corner from this region to this region, okay? And then what you have to do is subtract from the promoter region. Malab, if you are taking 1 kb upstream, so you have to subtract 1000 bases from the promoter region.
Exactly, ma'am. Exactly. And I tried doing exact the same process which you're saying, but the refseq sequence was not given
No,
for
no,
this gene.
it must be given. You must be searching from RefSeq. What you can do is you can check for a genomic sequence, but it's written like it must be written in front of genomic sequence, RefSeq genomic sequence. Or what you can do if you are still facing problem in it, you can make a Word file of it and can email us ki what steps you have done and what other problems you are facing.
Ma'am, is it possible to do it here right now? Means if you can show me,
Okay, ठीक है.
if I can give you my gene name, can you show me?
हाँ हाँ हाँ,
Because I
okay.
was facing an issue.
Okay.
Ma'am, I'm having a doubt in the same problem.
Okay.
Okay. Ma'am, how did you choose that HIF1 transcription factor?
What?
How can we choose one?
No,
How did you choose
that was
HIF?
sorry, that was just an example. In this case, if mom has not given you any of the transcription factor, you can randomly choose one of the transcription factor from that software I've told you. Or you can, instead of choosing any transcription factor, you can even clone that particular 1 kb region. I mean, you need not have any transcription factor.
Okay, we can design primer from starting to end.
Yeah, yeah, yeah.
Okay.
So is the screen visible now?
Yes, ma'am.
Tell me the name of your gene.
ECE2.
What?
ECE2.
Okay. Make this is mRNA and genome. Oh, there it is. Can you see? Primary assembly. Can you see primary assembly?
Yes, ma'am.
So down it's written a genomic. So this, if you click here, FASTA format, so you can, on the right corner, you can see from selected region from this to this, right? Hello?
Yes, ma'am. Yes, ma'am.
So you just have to take 1000 base pair upstream from this promoter region, start site. Okay? So what you have to do, copy paste this one and subtract 1000 from it. Suppose update view. So whatever sequence you are getting now is 1 kb upstream. Okay. So I think I have done some mistake. This is not 1 kb. 10 kb. This must be 1 kb. So this is your 1 kb region.
Okay, ma'am. Thank you so much, ma'am.
Okay.
Thank you.
गरिमा का जो टॉपिक है वो पहले बोल के पूछ रहे थे तो वो चल गया।
So anyone else, if anyone else having any doubt regarding this topic, you can tell me now.
So, ma'am, we can make primer by using like starting CAC, TTT from here only. We don't
You
need to
can
choose any transcription factor in that.
you can do that also. Other than that, if you wish to choose any transcription factor, then you can choose any. Supposedly you put your genomic the sequence into promo and it will give you a list of transcription factors which can bind to your sequence. So you can choose any of the transcription factor by yourself, or you can, if you don't want to clone your this transcription factor site, then you can choose to make primers from starting of the region to end of the sequence.
Okay, ma'am.
Any other questions?
Hello, ma'am.
Yeah, hi.
Ma'am, I had mailed you about that cloning in pet vector actually. So, uh, my confusion is actually that in the earlier vectors of the pet series, um, they mostly have a BAM-H1 restriction site ahead of the T7 um, promoter sequence, and my gene of interest also has a BAM-H1 site, so I cannot use that. And if I there are two or three more restriction sites which are present in my gene of interest. So if I try to clone it between XBA1 and NCO1, which are not present in my gene of interest, then the ribosome binding site comes in between those two. So it will not be economical to cut the RBS and then add another cosec sequence in it. So I'm not able to understand which two restriction sites to clone it in between, or choose another pet vector, because there are a lot of pet vectors in the series.
Which vector she's talking about?
Pet
Pet
vector.
vector. Which vector was it exactly? Pet 26?
No, I'm cloning it in pet 11D.
11B. Okay. So actually, we would need to see the map for it to be sure about this. Uh, should I share my screen? But there are other so pet 11A B. Sorry.
D, not D. D.
Not B.
Can you again repeat your question, please?
It is pet 11B. I put pet 11D. D for dog.
D.
D.
Okay. And can you please repeat the question? What is the problem you are facing?
There is a BAM-H1 restriction site present ahead of the T7 promoter,
Okay.
and my gene of interest also has a BAM-H1 site, so I cannot use that. and a couple of other restriction sites present ahead of the T7 promoter are also present in my gene of interest. So I think I wanted to ask here that we have to clone it ahead of the T7 promoter only, right? Because it will be used for expression of the protein.
डाउनस्ट्रीम ऑफ द promoter,
Promoter,
no?
Downstream.
Oh, down. Yes.
है ना? Where is the map? So,
back.
what do you actually want to do? Background? थोड़ा सा बताओगे?
Uh, Sir, we have to clone it in pet vector. We have to
Clone
design
what?
for cloning of
अच्छा, or your fusion protein or something like that?
yes, yes, histidine tag. We have to add histidine tag in the forward primer.
अच्छा, in the forward primer you need to add a tag.
Yes,
It's better to avoid. I mean, you cannot actually use any restriction enzyme which is present in your gene of interest. So first of all, forget that those enzymes whichever are present in have sites in your gene of interest. Then you look for the other enzymes, and then you are facing some problem.
yes, sir. Actually, if I try to clone it between, I think yes, XBA1 and
Hmm.
NCO1, then RBS is present in between those two sites. So the RBS will not be present, and we need RBS for ribosome binding so that protein can be expressed.
Like one
In the
option,
MCS
what I
region there
can.
is RBS. Are you sure?
This is T7 translation RBS which she's
Acha.
talking about. If you see NHE1 and NCO1,
Then you cannot
I
use those
think you
enzymes.
huh?
XBA1 and NCO1.
But why are you choosing BA1? You can choose NHE1 and NCO1, right?
Their restriction sites are overlapping, I guess, 250 and 55.
Oh, we are fine.
So Bam, it's fine.
Cannot use that pair. Absolutely.
What about this T7?
See, you have to try for other combinations of restriction enzymes. If you can't find any compatible, then you have to see other factors also now whether these two enzymes have buffer compatibility and not star activity and everything you have to keep into mind. And but yes, you are right. If there is RBS binding site in between and it's getting lost, then you cannot use those enzymes.
So can I
Look,
try it in any other pet vector? Because there are a lot of human series. So,
I think you should select another pet vector then.
what is given in your assignment actually? You can do
no,
it in any
no
vector.
specific. Yes, yes, no specific vectors mentioned. Just pet vectors written.
All right. So you try the other one. Even
I've
then, if
tried.
you're having, yeah.
Yes, I have actually tried a few of them ahead of in the series, like 2B, 3D, 4D, and all,
Uh-huh.
which I was facing the same problem. And later on, they have a 6S, 6X tag uh, already in the vector backbone. So I was not sure if I could use that one because we have to add it in the forward frame.
Okay. You do one thing. You write one email, or I mean, in a Word file, you just state your problem and send it to us. We will write back to you. Okay.
Yeah, with the maps, like attach
Yeah, yeah.
the vector maps also that you were using.
Because when you also need to see, I mean, in detail, no. Otherwise, just like that, we cannot say. Can you email it to us by today?
Yes, yes, sure.
So tomorrow we'll get back to you positively. When is your submission date?
30th.
30th. So by tomorrow we'll get back to you.
Sir, I
हाँ
took I took PAT 28A factor, and already there are two histech sites are present in that PAT factor. So whether I can take that vector or should I change vectors?
तुमको कुछ समझ आया?
She's asking pet 28A she's using.
Uh-huh.
So should she use that vector or change it?
Why to change it, Diksha?
Why do you?
Sir, already it is having two his tag.
But then you also have to include a histidine tag with your forward primer.
Yes, sir.
Then why are you using that vector? Because it was given in the question.
Yes, sir. That's what I'm asking. Ki, should I change this factor now?
Okay, that I think only ma'am can say. I don't know if she has set the questions.
Yeah, what is the?
Sir, I have the same doubt in it, sir. If there are two histidine tags present in that vector, can we
Uh-huh.
add another histidine tag with our forward primer?
No, it is
Will
not
it be?
smart to use that vector, right? Actually, okay, let us talk to ma'am, okay?
Actually, what I think is if the vector already has a his tag, then you don't need to specifically add it
But if the
using the
question
primer.
is you have to add it
Yeah,
with your primer,
but if the
what is it
question,
mentioned in the question? Can anyone of you like show or say read it out?
Ma'am,
Um,
actually, the histidine
Sir,
tags which are present, they are present in two different locations, and means they're present in such locations that if we choose a particular type of restriction endonuclease site, that histidine tag gets is either in the upstream region or comes in the downstream region. It would not get coded. So that is why we were adding one more histidine tag along with the forward primer, because the location of the histidine tags were a bit on the offside.
Okay, just we got the question from Pallavi. Design primers for cloning the coding sequence with a N-terminal stack in a page vector. So if your page vector already has, I think you are what you are referring to, it can be used as an N-terminal tag or also a C-terminal tag if both of them are there. So you have to smartly design in a way so that your protein has an N-terminal histidine tag. I don't think design primers for cloning the coding sequence with a N-terminal. Okay, so the question is a little ambiguous, like it is not clear whether she is referring to you have to put the histidine tag in your forward primer or use the vectors histidine tag to incorporate in your primer in such a way that the protein is automatically tagged in the upstream region or N terminal of your protein of interest. So this is a little unclear for me as well. Anuja, do you know what she is referring to?
so basically what the woman.
Hello, another just since it's not explicitly mentioned that this needs to be expressed. Like for the mammalian vector, ma'am mentioned that it was for expression in the mammalian vector. In
Correct.
this case, does do we have to clone it in such a way that it needs to be expressed as well?
Yeah. Idea नहीं है.
Can you repeat the question once?
So, like the previous question was the question about in was to clone it in a mammalian expression vector. So it was for expression. Now in this question for the pet vector, ma'am has not mentioned that it's just for cloning the CDS, not explicitly mentioned that it is for the expression of the clone CDS. So
Yeah, actually, pet vectors are generally for expression only. So, the idea of this is to have a fusion product, like a fusion protein, that is like your particular protein and the his tag attached to it. So, like a fusion product, right? So this basically is helpful when you're like trying to purify the protein later. Like you can use the his tag and then go for purification, like further downstream process.
So since this is a bacterial vector, so do we need to like in the previous question somebody was asking about the RBS being disrupted. So if that is happening, do we have to add a Schindel-Gardner sequence?
No, that is like already incorporated in the vector. So we don't need to specifically add.
Triangle.
Yeah. So But the only confusion here is whether we should select the vector which already has a histidine tag, or whether we should go for a vector which does not have a histidine tag, and we need to design our primer such that the forward primer has the histidine tag. So that is the only confusion, and for that
We will talk to ma'am and
yeah, we
write to
will
you.
talk to ma'am and then probably clear it out.
Ma'am, how do we design primers for reverse transcription PCR?
We design what?
For reverse transcription PCR.
What design what for reverse transcription feature?
Timers.
Primers. Diksha?
Yes, sir, primers.
Okay, so to detect a particular gene, you are asking.
Yes, sir.
Okay, so is there a question like in your assignment, or you're generally asking?
Yes, sir.
Can you read out the question?
Sure, sir.
Okay.
Design primers for reverse transcription PCR mediated detection of your transcript. One primer should be from intron-exon boundary.
All right.
And I don't have any intron in my gene of interest then.
No, actually, one primer should be from the exon-exon junction. Okay. So I'll briefly show you how to do it. Like, can you share your screen, Anuja? Give us any gene. What is your gene?
ECE1.
ECE1.
Yes, sir.
फिर गूगल में जाके EC अच्छा रेफसेक लिखो REFSEQ. Are you following?
Is it
Yes, sir. Yes, sir.
Okay, okay, just follow. the gene का नाम लिखो. One. Homo sapiens. For first वाला ही करो. Till here, did you follow?
Sir, I can see only NCBI home screen where. Yes, sir. Now I can see. Yes, sir.
Okay, okay. Now go to pick primers. Many ways to do it. You can manually identify introns and everything, or this is a easier way to do it, like NCBR primer blast. Here, a few things you need to follow. Like if you are designing primers for qPCR, then all right. Okay, okay. Go down. Here, there is one thing called exon intron selection. Can you see? यहाँ पे mouse लगाओ. Yeah. Here you need to select exon junction. Primer must span an exon-exon junction. Okay.
Yes, sir.
Okay. Then you go down. Yeah, right. And here database is RefSec mRNA. This is okay. Organism is 9606, which is human. And maximum target amplicon size is okay. And now you go upward. One thing here, PCR product size maximum you do it 250. For real-time PCR, you need to like make sure that your amplicon or whatever you are amplifying with your primers, it should not exceed more than 200 or 250 base pairs. Minimum is 80, maximum is 250. And the TM and everything else, we will keep it whatever there is. These are optimum conditions. Now just go down and click on get primers. Now this software will return to you a few primers and will also give you a map that this primer it spans which exon boundary. So it will give you a pair of primers. One of them will span an exon boundary. So in that way, and you check the other parameters like the TM and GC content and everything that you need to check for primer designing, and then you select the best one. Also select whether these primers amplify any non-specificities that you can check by giving the primers in the primer blast only, forward and reverse primer. And if you need to select the database from RefSec, you need to change it to genomic DNA and then click on find primers. It will show you whether these primers bind to any non-specific products other than your product of interest. But I think if you like design it from an exon junction, that is fine. So it sometimes takes some time, but it will give you.
Sir, but the question is asking one primer should be between intron-exon boundary.
It is the same thing. Exon junction and intron exon boundary is the same thing. You imagine that intron will be cut out, right? So automatically exon 2 will come and sit there.
Okay, okay.
Yeah. So it is the same thing. Sometimes it's confusing, I understand. See here. Okay. Can you see the screen?
Yes, sir.
Okay. All right. So here you can see the many exons 1, 2, 3, 4, 5, 6. So intron exon junction and exon junction are the same because the intron has been automatically cut out after like splicing. And here all the primers they have shown this software has returned is in the boundary. Primer 1 is the boundary of, or primer 2 I think is in the boundary of exon 2 and 3. So all of these primers are here. Here are the primers. Product length they are showing 210 base pair, TM, GC percentage, self complementarity, everything is given here. So you just need to make sure it is binding ECE1 transcript variant 2, and that's it. So it's easy, right? And here you just need to check which exon it spans. So you hover your mouse over here and you will find everything. You just write that this primer spans the exon 2 and exon 3 junction. Correct?
Okay, sir.
Okay.
So,
Yes.
so I have done the same thing that you have told now, but in my case, they are showing that no primers are available for such thing.
Ah. Then you have to like manually you can go and look for the different exons, exon intron what are there present, and then you can manually try to design it. But what is your gene of interest?
CXCR3.
CXCR3 should actually have. Anuja, but go back and change the gene of interest CXCR3. हाँ यहाँ पे CXCR3. इसमें click करो. तुम एक बार सर्च कर, मैं क्विकली एडिशन बफर डाल के आता हूँ.
नहीं वो तो जीन नहीं कौन सी जीन का करना है ये वाला हाँ हटाना है वही स्टेटस मेंशन खोलो तो गूगल हाँ इधर जाओ एक सेकंड दिखाऊंगी जाओ वो एनएम बोले उस पे जाओ तुम लोग का टीए चल रहा है क्या
Are you guys following?
Yes, ma'am.
Just
थोड़ा सा टाइम ले अब जाओ मैं देख लूँगा पता नहीं सर
तो भी खोलने का कुछ है जरूरत तो नहीं है पहले नाम से
Meanwhile, anybody else, any questions?
Sir,
Yes, yes.
sir, I have the doubt regarding the primer designing. Actually, when I'm designing the forward primer from the first nucleotide to up to 25 nucleotide, I am getting around 6 hairpin loop, and the TM is around the 70 degree centigrade for the highest one. But when I am reducing the length, then also I'm getting like minimum 1 hairpin loop, and the TM is 60 degree. But around the nucleotide 14th, then the hairpin loop is 0. But the 14
14 nucleotides
nucleotide,
you cannot take.
the range is not,
Minimum,
yes sir. So like in that sense, how would I generate the primer?
minimum you should take like 18.
Yes sir. So like
Now
how
see,
should I do
this
primer?
is your protein of interest, right? Right. So it is
Yes
not
sir.
possible to take from any other sequence. So one thing you can do actually, um, but I'll come back to your question. Who had this particular gene?
Sir, it's Miss Reoshi Karmakar.
Srevashi. So it is showing some primers I can see. What was in the problem in your case? I'm not sure.
Okay, so actually I have clicked on the whole gene and ma'am has clicked on a transcript variant. That's why.
No, you need to detect the transcript variant, right? So you'll have to click on the transcript variant one.
Okay, sir.
So here I can see at least 10 primers they're showing. This particular gene has only 2 exons. Okay. So obviously the forward primer will be in the junction of this exon 1 and exon 2. And there are varying length of your PCR product, so they have designed multiple primers based on that. So, so I think that answers your question, Sreyashi. And who was having problem with the forward primer?
Sir, it's Miss Reoshi. Actually, that time I got it wrong.
Okay,
So
okay.
that was.
Oh, sorry. Okay. So the thing is, you have no other option but to select the forward primer from like ATG. It starts from ATG, right? What you can do is you can take a few bases upstream of ATG, but only a few,
Okay, sir.
like 1 to 3 bases, and reduce a few bases from the, I mean, 3' end of your primer. So see, so what will happen is it will clone that a few bases upstream of your ATG also. But obviously your translation will start from ATG, right?
Yes, sir.
So in that sense, that should not be a problem. But if you are doing it for a fusion protein, then you have to be very careful that this histidine, wherever it starts, if the translation starts from histidine, which is in the N terminal of your protein, then you need to make sure that all of this is in frame. But if your in frame, you understand, right? Because in codon, like pairs of 3, the codons
Yes,
are
sir. Yes, sir.
So you have to make sure that your ATG falls in frame. You understand my point? Because if your ATG does not fall in frame, the protein will not be made, like methionine will not be made. So,
Sir,
and
actually same problem I'm getting for the reverse primer also.
okay.
In that sense, how could I do that?
See, one thing is
And sir, for reverse primer, I'm getting the TM is around 30 degree. So like
30 degrees
that is
still
very
work.
much.
30 degrees might still work, but
But
your
sir, I mean, for me, the forward primer is coming, the TM is 58, and like reverse primer, that is the 30. So like difference is huge. I mean, it should be in between 3 degree to 5 degree.
oh, you are saying like TM difference. All right, all right.
Yes, sir.
I was
Yes, sir.
thinking that the hairpin it is generating, it has a TM of that 30 degree.
No, no, sir.
No, no, no. Then see, must be there. The GC content must be very different in the forward region and the reverse region.
Yes, sir.
Okay. So in the reverse primer case, you can take like you can go beyond the reverse primer because in the reverse primer you will obviously add the stop codon.
Yes, sir.
So the stop codon will stop the translation. You can go beyond your sequence of interest and take a few bases to adjust the TM so that they can come close. Even if then it is not being adjusted, like you have to make it close, right? Forward and reverse, otherwise at least 5 degree temperature aspas. So it should be there. Otherwise there is no point.
Sir,
So
if the amplicon size then increase up to 500 base pair, then
500 base pair. What is your amplicon size usually normally?
sir, it should be 250 to 300.
Oh, now see, now I am getting confused whether you are cloning a coding sequence or you are doing real-time PCR primers designing.
So that is for the CDS region.
So for the
I'm
CDS
designing the primer. Yes,
region,
sir.
there is no boundary, like it should be like this basis only, because proteins can be like thousand, I mean 10 kb long sequences also people clone. For the CDS region, there is no rule. Okay, whatever the
Okay,
size
sir.
of the amplicon is, you have to clone that only. But for real-time PCR detection primers, the amplicon size cannot be more than 200 or 250 bases. That is what is recommended. You try one more time. See, so your forward primer will start from ATG, and you have to take minimum of 18 bases. For the reverse also, you try to take 18 bases, or if the TM is lower, usually so much difference does not happen, Shreyashi. I would suggest you to go ahead and check once more. You are telling
Sir.
50 and 30, so there is huge
Yes,
difference.
sir. Sir,
Until
I
and unless
checked
your
it.
reverse
I
primer
mean,
all
more than 3
80,
times.
okay, all 80 is there. Is that it?
Yes, sir. That's why the team is decreasing.
Okay. So when you are selecting the CDS region from the NCBI,
Yes, sir.
uh, you actually copy the whole FASTA sequence, even beyond the CDS region.
No, I am just taking the CDS region
No,
only.
no. What I am suggesting you to do is
Okay,
you
sir.
take
Okay, okay.
even beyond the few base pairs of the FASTA sequence, and you try to make the reverse primer from beyond your CDS. But make
Okay,
sure
sir.
the stop codon is present. You cannot ignore that. So when it will encounter the stop codon, I mean, obviously stop codon will be present. You do not need to make the stop codon in the reverse primer again, because when translation will start from the start codon, it will go, and whenever it encounters the stop codon, it will stop. Right. So what I'm trying
Yes,
to
sir.
tell you is you go beyond your CDS and try to design like see whether the GC content is coming closer and your TM is coming closer or not. No problem in that sense.
Okay, sir.
Even then, if you are finding problems, then you state that you mention that in your primer, whatever you closest you get, you design it, and you state I have checked multiple places, but the TM difference is too high, could not, so we will consider that and your marks would not be deducted. If you give a
Okay, sir.
justification about it, I have checked multiple sites and there is no other option. This much difference is there. Then your marks won't be deducted.
Okay, sir.
Okay.
Thank you, sir.
Okay.
Sir, while designing primers for reverse transcription PCR, why do we keep our transcript length 250 base pair?
Because see, in real-time PCR, in normal PCR, you usually give an extension, right?
Yes, sir.
Yeah, but in real-time PCR, you need not give an extension, which also saves time and very quickly you can detect multiple genes. And it is not necessary to go beyond, so much big length because if your one primer is from an exon-exon junction, it will obviously not detect any genomic DNA contamination, first of all. And secondly, if you are done in 30 seconds, see, it is an annealing and extension combined. Whatever time you give it for annealing, it will automatically extend for that 200 300 base pair. So obviously it is more economical and what should I say, smart in that sense. So why should I go for like 500 600 bases?
Okay, sir.
It is not theoretically wrong, you can go, but your PCR will be longer, like 3 hours 4 hours.
Okay, sir. Okay.
Yeah. Okay.
Sir, when I was designing the primer, so the GC content was coming out to be 70% in the
70%.
yes, sir.
Which one, forward or reverse?
Forward.
Forward. Okay. See, it is not impossible to clone with a 70% forward primer. You check it and you mention it key. Since this is a forward primer, you have no other option. There are
Okay,
some
sir.
polymerases which have you might write one sentence that this might require the addition of DMSO or a GC rich buffer for amplification. Since this is there was no other option and my forward primer has 70% GC content. Sometimes you have to work with the 70% GC content also.
So what is the purpose of DMSO addition?
Oh, DMSO, when see that if there is a GC rich, your amplicon is GC rich, for example, more than 60% GC if it is present in your amplicon, DMSO helps in denaturation. It will bring down the TM a few degrees and your amplification will be smoother. You will not see like smears on your PCR product and other things. So DMSO basically helps in denaturing. Whenever your amplicon or your primers are GC rich, it is recommended that you use DMSO, 3% DMSO to start with in your PCR master mix. So that will help to basically G and C binds with like triple hydrogen bond. The bond is stronger, it is not easy to open that up. That is why it is suggested that, and if it is very loose, the PCR product it can bind anywhere and it is like non-specifically it can give amplification. So you compromise in your like specificity and your like what should I say? Reaction optimum conditions. So in that sense, DMSO is added to bring down the TM a few degrees. So you can write that is important note since this is a forward primer and I could not change its TM because obviously you have to take the from ATG at least 18 bases. You can tweak around the number of bases and see if the TM comes down a bit. Even then, if you are finding problems, you just write an explanation about whatever problem you are facing. If your explanation makes sense, then obviously marks will not get deducted.
Sir, actually the TM is fine. It's around 48 degrees.
Oh,
Like,
then
yes.
that's okay.
Okay, sir.
Somebody is raising hand. Who is it?
I want to know like how do we design the SH RNA? So like I studied about how to design SI, but I couldn't understand how to design SH RNA.
shRNA TA has not been conducted yet.
Yeah, it has not been conducted, but it was mentioned at the time, and so I was just asking for a like a small protocol or like description
No,
if
no,
we can get.
we will discuss it in details.
Okay.
Probably in the next class. Okay.
Sir,
Yes.
sir, we have to submit the assignment before 30, on
All
or before 30.
okay.
And this is the question. It is the part of the assignment,
Yeah,
SIRNA part.
questions on siRNA and shRNA both.
Really? What
Yes,
question?
Yes,
sir.
ma'am.
Sir, I have submitted incompleted
You
till the SI part. So do I have to submit it again with the remaining part?
Hold on, hold on. I think we need to talk to ma'am, right?
Yeah, we'll confirm with
Yeah,
ma'am
yeah.
once.
Anuja will confirm with ma'am and write in this group. I think 5-10 minutes after the meeting. You don't leave and you'll get to see. Okay. Because if it is, has not been covered, it should not come in your assignment, right? Let's see. Anything else?
Uh, uh,
Sir.
One thing, sir, if it will not come in the assignment, I have already submitted till the primer parts. So do I have to submit again or it's okay?
See, that we will mention in the chat. You do not need to work on the siRNA or shRNA part. But we cannot say that. Obviously, ma'am has to say it. So we'll get in touch with her and write in the chat. So I don't think you need to resubmit it if it is not required. Those two questions.
Okay.
Shilpi was
Sir.
asking something. Yeah.
Yes, sir. So actually, while designing the primers for the cloning of PDS region, in my forward primer, I am getting a hairpin loop, and the TM of the hairpin loop is showing to be 0. So what does
Then it's
that
fine.
mean?
You don't have to worry about it.
Okay, but what is the meaning of TM 0?
It means
Yeah, I had mailed you also regarding this. So basically, it means that the structure is not stable. So you don't have to like really worry about the secondary structure which is not.
it
Okay,
means at very low temperature it
It
breaks
will
down.
melt, yeah.
Yeah.
okay,
Most
very good.
likely it will have like 80, 80, 80, which will automatically break down at very short stretch of it.
Okay, okay,
Sir, hello, sir.
Yes, yes.
Sir, I got confused with one question, which is provide the sequence for forward and reverse primers for cloning of 1 kb region upstream of the transcription start site in
Uh-huh.
PGL3 basic vectors. Is it the same questions designing the primers for promoter regions, which will be binding with the transcription factor?
Yeah, I
This is the
think.
same question, right?
Yeah, I think
Yeah,
it's
it
a, yeah.
should be the
Actually, see,
same.
the transcription factor upstream site in some how many bases it is mentioned? 1 kb. So the
Minus 1 kb,
yeah, yeah. So the promoter
minus
can
1
be
kb region of the upstream.
so the promoter can be situated in anywhere up to like 10 kb of your transcription start site upstream of it. So in some
Yeah.
sense you are right ki, it is the promoter only. So basically, I think you have to do
Okay,
it in the same fashion.
okay, sir.
Okay.
Okay, sir. Thank you.
Anyone else? Anybody?
I have one more question. So one question was that I'll just read out the question. Provide the sequence of primers for mutating any three base pairs in the construct that you had cloned in the PGL3 basic vector.
It is also in your assignment,
Yeah, this is also in the assignment.
but your SDM has been covered. Has it been covered? I don't know.
It has been covered.
Side
This has
directed mutation.
been
Oh, sorry. Okay, then go forward. Tell me.
Yeah, so I'm just sharing the question. So once we have it, once we have the clone ready,
Ah,
uh, what considerations go into this site-directed mutagenesis? Like how many base pairs or AT versus GC?
Oh, ATGC, I have not taken this TA, but I'll try to answer your question.
Because
Uh,
I don't think this was covered as yet.
yeah,
Was it?
Can you repeat the question?
it is
I
cited in
put it
GC sir.
in the chat box.
Chat, chat.
Uh,
Astha was supposed to take it.
Rahul sir, is it there in the meeting?
Rahul is there. Rahul.
I have taken two lectures so far. One is regarding the GFP fusion construct, and the second one is about transfection using lipofectamine.
Sir, SDM has not covered yet.
All right.
So
So,
just to sum it up, question number 5, 6, and 7 out of the 8 questions have not been covered as yet.
okay. Do you have the question?
Can
So
somebody share the assignment with us, like the question paper?
Yes, ma'am. Send.
Just tell us which one of these have not been covered, okay?
question 5, 6, and 7.
Okay. Uh, did anybody given attachment or something?
Can you attach the question paper also so that we know which questions they are exactly?
So I have actually overwritten my solutions in that. So if somebody has it has it free, then maybe they can.
Somebody can attach the question paper and we'll just go through that and see.
I'm sending it. You can check the chat box.
Okay, so fifth, sixth, and seventh questions have not been covered. So we'll ask ma'am and then we'll confirm with her once and we'll get back to you guys. Meanwhile, are there any other questions? Does anyone have any other questions?
Hello, ma'am.
Hello?
Hello,
Yeah. Yeah.
ma'am. For the eighth question, which is design the primers for the RT-PCR. You have just
Designing
mentioned about the primer blast uh, for designing the primer for RT-PCR. Am I
right.
correct, ma'am? Okay, ma'am.
Can you repeat?
The-
Your
Your
voice is
voice is breaking.
breaking.
Hello? Hello?
Yeah, Vibhuti.
Yes, sir. Sir, for designing the primers for RT-PCR, you have just mentioned about the primer blast.
Yes, yes.
Am I correct, sir? This is
Yes,
the
yes.
way to design the primers for RT-PCR?
Yeah, I suggested primer blast, yes.
Okay, okay.
But
Okay, sir.
there are other ways to do it also. You can
Okay.
manually find out its sequence and you can like select forward primer and then go ahead 200 whisper and select your reverse primer.
Yeah.
Or also there are other tools like IGT oligo analyzer, primer
Uh,
3. all this, you can go ahead and try for real-time PCR primer designing.
Okay, can we use Snapgene Viewer for designing the primers?
Snapgene viewer, yes, but you have to know which is the exon junction. Then you can use it. Like Snapgene also you can use. But viewer, I don't know if viewer has an option for primer designing because I don't use the premium version. But definitely you can use it to design. But the
Okay, sir.
condition should be met that one primer, either your forward or reverse, should span an exon or exon intron, whatever you'd like to call it, junction.
Okay, sir. Thank you, sir.
Okay.
Are there any other questions?
See, after this meeting, even still, if you are having any doubt, which is not clear or you are facing some problem, just write to, I think you can write to Anuja. We will forward it to whoever concerned, the TA who has taken it, and hopefully you'll get an answer back quickly. Okay, so if anybody else do not have any questions, I will close the meeting. We'll notify you about the questions that have not yet been covered. So Rahul sir is also there, Rahul Gupta, your TA. If you have any doubt from his part, you can ask him, like transcription and which one else? GFP fusion protein. About all this, you have if you have
Yeah,
any
if
questions.
you have any doubts from the practical on transfection and the second one, which was about designing a fusion EGFP construct, then you can ask me. Okay. Any questions?
Okay, so I don't think they have any questions. So we'll close it then the meeting then, and we will get back to you about the questions that have not been covered. Okay. Probably